Salve. In this video, I'm going to show you how to draw a generalized eukaryotic cell, specifically an animal cell. So according to the IB syllabus, you need to be able to not just recognize and label these diagrams, but you also need to be able to produce them. So I'm trying to show you how to produce them. Obviously, not every animal cell is the same. So your heart cells are different than your liver cells are different than your nerve cells. But this drawing is showing you the structures that are characteristic of animal cells. So because animal cells don't have a cell wall, I'm just going to draw kind of a circle shape. It doesn't have to be perfect. Animal cells are squishy. And then the only reason that I'm using pen is that the document camera can't really see my pencil. So you should be drawing in pencil. That's true for any IB diagram. On your actual IB test, you can use pencil for diagrams and charts and drawings, and I really, really encourage you to do that. So practice that now. I'm going to do an outline here in the middle, which I know you can't see very well, so I'm going to go over it in pen. This is going to be the nucleus, and something that's really important is that the nucleus has a double membrane, and it has these little open spaces called nuclear pores. And when we start to learn more about protein synthesis, you will learn why that's so important. Basically, the information in the nucleus has to be able to leave the nucleus and get to the ribosomes. So I'm going to finish up that double membrane here. You should do the same. So again, this is the double membrane surrounding the nucleus, and the little gaps are called nuclear pores. I'm going to add this nucleolus, or nucleolus, they're both fine pronunciation. That's the region of the nucleus where ribosomal RNA is produced. I'm going to add some crazy squiggles here. Your squiggles don't have to look the same as mine. I hope you can recognize what that is becoming. And then I'm going to do a similar thing over here. Again, I hope you recognize what we are drawing. Okay, that looks pretty good. I'm going to add this structure, which kind of looks like a stack of flattened pancakes. Some discipuli have told me before it looks like a Wi-Fi symbol. So whatever you think it looks like. And then I'm going to add some vesicles here. So I hope you realize what I've drawn is the endoplasmic reticulum. This is the Golgi complex, or the Golgi apparatus. That's the same thing. And then I drew these vesicles in here because there are vesicles that bud off of the endoplasmic reticulum, and then they fuse with the structures of the Golgi apparatus. The substances inside those vesicles get modified, and as they move through the Golgi apparatus, they're further modified, and they're often exported. So this vesicle might be heading towards the plasma membrane here to export something from the cell. I'm going to draw a lysosome here. So without being able to sample the chemical contents, we don't really know that that's a lysosome. I'm just telling you that it is. What defines it as a lysosome is the presence of digestive enzymes. So a lysosome is a vesicle that contains digestive enzymes. And its role is to break down sometimes food that's ingested by the cell, but often cellular organelles that are old or damaged. The cell actually breaks down its own old and damaged parts. Okay. A real cell would have more than one of these, but I'll just draw one. I'm drawing one mitochondrion. The plural is mitochondria, singular mitochondrion. And just like the nucleus, it's really important that this structure have two membranes. So those folds that I'm drawing are called cristae or cristae. But what you need to realize is that this is one membrane, and then we have an inner membrane. And when we learn about cellular respiration, we'll learn more about the importance of that. Okay, I'm pretty happy with my drawing. I need to add some dots. I hope you recognize what those are. If you do, try to remember what makes these different from prokaryotic structures of the same type. I hope you remember that these are 80S ribosomes. 80S ribosomes. Okay, so if you turned in this beautiful picture on your IB exam, or for an exam in this class, you would get 0 points because it has no labels. So we must add our labels. I'm going to label the nucleus. I'm going to label a lysosome. Oh, I'm going to extend that line, lysosome. This line here is representing the cell membrane or the plasma membrane. They're the same thing. You can write either. Plasma membrane, plasma membrane. So the plasma membrane controls what goes in and out of the cell. And then this internal area of the cell is full of this jelly-like liquid called the cytoplasm. And the cytoplasm is what all those organelles are resting in, so the cytoplasm. Here's our friend 80S ribosome. Because this is just one, I label it mitochondrion. And again, the plural, mitochondria. This series of stacks here and the associated vesicles is the Golgi apparatus. Or the Golgi complex or the Golgi bodies, it's the same thing. I'll label one of these as a vesicle. A vesicle is just that small membrane-bound sac. And then I hope you notice there's a couple things left. So these two structures are similar, but because this one has ribosomes, it's called the rough endoplasmic reticulum. You can also call it the rough ER, and this is the smooth endoplasmic reticulum, or smooth ER. A couple of people labeled this as a soft ER. That's cute, but it's not what it's called. It's the rough and the smooth ER. I'm not going to draw a vacuole because vacuoles in animal cells tend to be temporary, so they're not necessarily a defining characteristic of animal cells. So I'm gonna leave it there.